Okay, so welcome to this next video in which we are discussing VIP and PACAP receptors. Okay, so we're in the process of discussing Gprotein coupled receptors and then the heterotrimeric G proteins which they interact with. We've discussed that heterotrimeric G proteins consist of an alpha subunit, a beta subunit, and a gamma subunit. What we now want to discuss is the diversity of heterotrimeric G proteins, because there is not just one alpha subunit, there is not just one beta subunit, and there is not just one gamma subunit. Okay, so I now want to tell you about the different types of alpha subunit, the different types of beta subunit, and the different types of gamma subunits. Okay, right. So we'll begin then with the alpha subunits, more correctly called the G alpha subunits, okay, to denote the fact that we're talking about an alpha subunit of a heterotrimeric G protein. Okay, right. So there are 16 different genes for alpha subunits of heterotrimeric G proteins, okay, and these produce in total 21 different subunits. Now, this looks like a little anomaly at first, until you remember that some genes can have multiple splice variants, okay? So certain genes can make more than one protein, and these different proteins are known as the different splice variants of that gene, basically. Okay, right. So, let me just remind you of the central dogma of biology to explain this to you in a little bit more detail. So, let's let these two parallel lines here now represent a piece of double-stranded DNA rather than the phospholipid bilayer. Okay, and let's say that this region that I'm now boxing is a gene for an alpha subunit of a heterotrimeric G protein. Okay, I'll even colour it in. So this is going to be a gene. Now, one of the strands of this double-stranded piece of DNA will be the coding strand of the gene. Okay, so I'll colour in the coding strand in purple here. So this will be the coding strand. Now what do I mean by that? Well, I mean that it will be the strands that RNA polymerase II actually works its way along to produce a piece of RNA that is complementary to that. Okay, so this is the strand that RNA polymerase II will work along. And RNA polymerase II is often abbreviated to RNAP2, like so. RNA for RNA, P for polymerase, and then 2 for 2. Okay, so RNA polymerase II will work its way along the coding strand and produce a piece of mRNA. The other strand that is not used by RNA polymerase II, which I'll now colour in green here, okay, the complementary strand to the coding strand, this is called the non-coding strand. Okay, right, so let's say we have here a gene for some alpha subunit of a heterotrimeric G protein. RNA polymerase II will work its way along the coding strand and produce a piece of mRNA that is complementary to that coding strand. Okay, so the little line I'm now colouring here in orange here, this is the piece of mRNA that's complementary to the coding strand. So here we have it here. Now, this piece of mRNA that you produce is not ready to go for a ribosome yet, okay? So you cannot just translate the primary transcript that you've made by producing a piece of mRNA that's complementary to the coding strand. This is called a piece of pre-mRNA. And the reason you can't just translate this is that there are portions of this pre-mRNA that are not supposed to be translated, and if you did translate them, you'd end up with a rubbish sequence of amino acids which would be non-functional most likely, and probably would aggregate into amyloid plaques and would be very bad. Okay, so only certain portions of this pre-mRNA are actually supposed to be translated. Okay, so I'll box these portions here. So let's say that these 3 portions that I have now outlined in boxes here are the 3 portions that are supposed to be translated. Now the portions that are actually supposed to be translated are known as the exons. The portions in between the exons, which are the junk portions, okay, these portions are not supposed to be translated. These portions are known as the introns. Okay, so what has to follow is a post-transcriptional modification where we're going to cut the pre-mRNA up and we're going to cut out the introns and then re-sew together the exons to make a smaller piece of mRNA, okay, which consists now of just the exons put together basically. So here in orange, here's the smaller piece of mRNA, and this now is ready to go for a ribosome. This is called the mature mRNA, and the process of cutting out the introns and then sewing the exons back together is known as splicing. Okay, so we splice the pre-mRNA and produce a mature piece of mRNA. Okay, now there are multiple ways of splicing a piece of pre-mRNA, and these different ways of, well, in some cases I should add, in some cases there are multiple ways of splicing the pre-mRNA, and this can result in different mature mRNAs. Okay, so let me give you a very easy to understand example of this. Let's say that this little exon in the middle here is not actually essential. Okay, so you can either have this exon here in your mature mRNA, or you can leave it out basically. So if you have an exon like so, which is optional, then you can produce two different pieces of mature mRNA from this piece of pre-mRNA. You can either produce a mature mRNA when you have all three exons making up the mature mRNA, or you can produce the one where only the first and the last exon are within the mature piece of mRNA, and you've cut out that middle exon as though it was an intron basically. So you'd have a long mature mRNA which contained all three exons, and a short mature mRNA which contained just two of the exons. So I'll draw this here. Okay, so here's the short one. And these now are two different pieces of mature mRNA that you have produced from the same piece of pre-mRNA. Okay, so from different copies of this pre-mRNA. I'm not saying that you take this one piece of pre-mRNA and somehow end up with both of them, no. You take two different pieces of pre-mRNA, splice one in one way and you get a short one, splice the other in a different way and you get the long one. Okay, so you can produce there are two options available for you as to what the pre-mRNA can become basically. And both of these will now code for different proteins. Okay, this one will be a longer protein and this one will be a shorter protein. And these different proteins are now called the different splice variants of that gene. So this is how one gene can produce multiple different proteins through the variation in splicing of the pre-mRNA basically. Okay, so that's how 16 different genes for alpha subunits of heterotrimeric G proteins can give rise to 21 different subunits for alpha subunits of heterotrimeric G proteins. Now, we're not going to dwell on the different splice variants of the alpha subunits. We are instead going to look at the 16 different genes. And the reason is the different splice variants of the same gene often have very similar functions anyway. So we're just going to focus on the 16 different genes for alpha subunits of heterotrimeric G proteins. Okay, right. So the alpha subunits of heterotrimeric G proteins, the genes for them anyway, are divided into 4 major families to help us understand this. Okay, so there is the G-alpha S family of alpha subunits of heterotrimeric G proteins. That's the first family. Then the second family is the G-alpha I/0 family of alpha subunits of heterotrimeric G proteins. The third family is the G-alpha Q/11 family of alpha subunits of heterotrimeric G proteins. And finally, there's also the G-alpha 12/13 family of alpha subunits of heterotrimeric G proteins. Okay, so let's begin with the G-alpha S family of alpha subunits of heterotrimeric G proteins. Okay, so there are 2 members within this family. There is the G-alpha S gene itself, and then there is also the G-alpha OLF gene. Okay, now G-alpha S has many different splice variants, okay, and G-alpha OLF only has one splice variant. Now, both of these 2 two alpha subunits of heterotrimeric G proteins, once they are in the on state with GTP bound, they activate adenylyl cyclase enzymes, okay, and we will see G-alpha S very shortly because this is the one that's downstream of the VIP PACAP receptors. Okay, but the reason both of these are put into the same family is therefore because they have very similar functions. In fact, G-alpha OLF seems to be the equivalent of G-alpha S in certain cells of the body. So in certain tissues of the body, such as the olfactory epithelium, which is why this is named OLF for olfactory epithelium, you find G-alpha OLF expressed instead of G-alpha S. So in certain tissues they have very low levels of G-alpha S, and instead they have G-alpha OLF, and it seems as though G-alpha OLF merely replaces G-alpha S in some places, basically. But most tissues have G-alpha S and not G-alpha OLF. Okay, so they seem to do pretty much exactly the same thing, but they are separate genes. Okay, right, and they're both within this G-alpha S family of alpha subunits of heterotrimeric G proteins. Moving on to the G-alpha I/0 family of alpha subunits of heterotrimeric G proteins. This is more difficult. This one's got absolutely loads of members in. Okay, so firstly it's got the G-alpha I alpha subunits of heterotrimeric G proteins, and there is not just one of these, okay? So if you're seeing this for the first time, this might be a shock, but there is not just one G-alpha I subunit. Okay, there is in fact 3 different genes for G-alpha I subunits. There's the G-alpha I1, the G-alpha I2, the G-alpha I3. Now, they pretty much all do the same thing. So people will often say that they are dealing with a G-alpha I alpha subunit of a heterotrimeric G protein, and what this means is it's one of the 3. So often people don't know specifically which one they're dealing with, or it could be the case that they're dealing with all 3. Okay, so just to generalize it, they'll say I'm dealing with a G-alpha I alpha subunit, or my G protein coupled receptor works through heterotrimeric G proteins with a G-alpha I subunit within them, and that means one of these 3 subunits. Okay, but they are 3 separate subunits that are coded by 3 separate genes, and they all have very similar functions, and they're all in the same family. Now, this family also contains a gene called G-alpha O, which is the other one after which the whole family is named. This, again, has very similar functions to the 3 G-alpha I alpha subunits of heterotrimeric G proteins. Okay, so the G-alpha I subunits tend to inhibit adenylyl cyclase enzymes, certain adenylyl cyclase enzymes anyway. They don't inhibit them all, actually, as we'll come to later. G-alpha O also has the potential to inhibit adenylyl cyclase enzymes. So again, very similar functions, which is why they're all grouped into the same family. G-alpha O has 2 splice variants, which if you read the literature, you will hear about all the time. So I'll give you their nomenclature. So there is G-alpha OA and there is G-alpha OB, which are these 2 different splice variants produced from this G-alpha O gene. Okay, right, and they, again, the 2 splice variants pretty much have the same function. Right, so, next up, another gene within this G-alpha I/0 family of alpha subunits is G-alpha Z. Okay, and again, this has similar functions to these 4. Again, it seems to inhibit adenylyl cyclase enzymes. So these 5 all are capable of inhibiting certain adenylyl cyclase enzymes. And when we get on to discussing adenylyl cyclase enzymes, I'll come back to that, or I'll mention it again. Okay, now, the next 2 are very, very different from these 5. Okay, these 5 all inhibit adenylyl cyclase enzymes and thereby reduce the production of cyclic AMP. Okay, the next 2 alpha subunits are called G-alpha T1 and G-alpha T2. Okay, and these 2 subunits do not inhibit adenylyl cyclase enzymes. Instead, they activate phosphodiesterase enzymes and they make cyclic GMP go down within the cytoplasm. So these make cyclic AMP go down by inactivating the production of cyclic AMP. These make cyclic GMP go down by activating the destruction of cyclic GMP. Okay, now, why are they known as the G-alpha T's? Okay, and collectively they would be called the G-alpha T's. Okay, well basically it's because these alpha subunits are found in heterotrimeric G proteins that are called transducins. Okay, so we go back to the pathway within photoreceptors in the retina. So we've already discussed that in rod cells, at the very least, the G protein coupled receptor for light is rhodopsin, and you have a very similar one called iodopsin in cone cells. Okay, now when light activates these G protein coupled receptors, they must activate a heterotrimeric G protein. Okay, the heterotrimeric G proteins they interact with are known as the transducins. Okay, so they consist of an alpha, a beta, and a gamma subunit. Now, you have different transducins in rod cells and cone cells. So you have rod transducin, which is the type of transducin in rod cells, and you have cone transducin, which is the type of transducin in cone cells. And the difference is that the alpha subunit of rod transducin is G-alpha T1. Okay, so this is the alpha subunit of rod transducin, and the alpha subunit of cone transducin is G-alpha T2. Okay, so these two are the alpha subunits of rod and cone cell transducins, respectively, and when light hits the G protein coupled receptor within photoreceptive cells, it will activate these rod and cone cell transducins, and then you'll get the activation of these alpha subunits, which will go off and inhibit phosphodiesterase enzymes, causing cyclic GMP levels to go down within the cytoplasm of the photoreceptor, and this then continues on the pathway of transduction of the visual stimulus. Okay, so those two are put in this family with these five because they again make cyclic nucleotides go down. The final member of this family of 8 alpha subunits is the G-alpha gust alpha subunit, which is involved in the gustatory system. Okay, so it's involved in the transduction of gustatory stimuli. Okay, right, so those are the 8 different genes within that G-alpha I/0 family. Within the G-alpha Q/11 family of alpha subunits of heterotrimeric G proteins, then, there are 4 members. Okay, now the most famous member of this family is G-alpha Q itself, but there are then 3 other alpha subunits of heterotrimeric G proteins which have almost identical functions to G-alpha Q. Okay, so the first one is the other one after which the whole family is named, which is G-alpha 11. Then you also have G-alpha 14, and finally G-alpha 15/16. Okay, now G-alpha 15/16 is a single alpha subunit, even though its name suggests that somehow it's 2. So I'll tell you the story behind how it became called G-alpha 15/16 so that you understand that this is only one gene for an alpha subunit. Okay, so basically the people, well, there were 2 groups, there were 2 different groups who were working on alpha subunits of heterotrimeric G proteins. Now one group worked in mice, and they found a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 15. Okay, then another group was working in humans, and they found a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 16. Okay, so this was in mice, and this was in humans. Okay, later it transpired that actually these 2 different alpha subunits, one that had been found in mice and one that had been found in humans, were actually equivalent. Okay, so this was this in mice and this was this in humans. Okay, so there was then the movement to rename them G-alpha 15/16 because we don't want, we don't want a gene being called one thing in mice and another thing in humans. So we wanted to rename both of them G-alpha 15/16. So we wanted G-alpha 15 to be called G-alpha 15/16 in mice and G-alpha 16 to be called G-alpha 15/16 in humans. Now it hasn't caught on as well as we'd hoped. The people working in mice still call it G-alpha 15 and the people working in humans still call it G-alpha 16. But it would be nice if you used G-alpha 15/16 to show winning. Right, so all of these four alpha subunits then pretty much do the same thing. They all go off and activate phospholipase C beta family enzymes, okay, and that leads to the breakdown of phosphatidyl inositol 4,5-bisphosphate into inositol 1,4,5 trisphosphate and diacylglycerol, which then leads to the release of calcium from the ER, and then the calcium and the diacylglycerol together activate protein kinase C enzymes. Okay, so these four all do the same thing. Even if you'd thought that G-alpha Q is the only one that did that, these three also do pretty much the same thing, which is why they're in the same family as G-alpha Q. Okay, right. Finally, there's the family that no one has ever heard of. Okay, which is the G-alpha 12/13 family, which contains two alpha subunits. It contains G-alpha 12 and G-alpha 13, and both of these are involved in cytoskeletal remodeling. Okay, right, so there we have it, all the different alpha subunits of heterotrimeric G proteins. Okay, so we've now covered the scope of different alpha subunits. Okay, we haven't gone into all the different splice variants, but the different splice variants of some of these genes do pretty much the same thing, and people rarely ever distinguish between them. Okay, right, G-alpha O is the exception. People often do distinguish between the two splice variants of G-alpha O. Okay, so next up, let's talk about the G-beta subunits. Now, this shouldn't take us long. Okay, so G-beta subunits. There are 5 known genes for G-beta subunits, and these code for a total number of 6 known subunits. Okay, now, we understand completely what's happened here. One of those genes must have 2 different splice variants, which is how we have 5 genes and yet 6 subunits. Okay, so that's indeed what has happened. So, the 5 genes then, we'll start with the 5 genes. They're called G-beta 1, G-beta 2, so it's a very nice simple naming system, G-beta 3, G-beta 4, and then finally G-beta 5. Okay, right, so that's the naming system for the 5 different beta subunits. Okay, next up we need to discuss the one that has 2 splice variants. So G-beta 5 is the only one that has 2 splice variants, and the 2 splice variants are called G-beta 5,5 beta subunit and then the G-beta 5L beta subunit. So those are the 2 separate proteins that you can get from this same gene. All of the others only have 1 known splice variant, and therefore their corresponding subunit is known as the G-beta 1 subunit, the G-beta 2 subunit, the G-beta 3 subunit, and the G-beta 4 subunit. Okay, so that's G-beta subunits of heterotrimeric G proteins. Next up, let's have the G-gamma subunits. Okay, right. So, how many different genes are there for G-gamma subunits? Well, there are 12 different genes known for G-gamma subunits, and these code for a total of 12 known subunits. Okay, so there are no splice variants here. Each gene just has a single subunit, and the naming is almost perfect, but it has 1 blip in it. Okay, so the naming then, we have G-gamma 1, then G-gamma 2, then G-gamma 3, all the way up to G-gamma 5, and then there's a blip. Okay, there is no G-gamma 6, and we move straight on to G-gamma 7, and then you continue on all the way up to G-gamma 13. So because you missed out G-gamma 6, you now have to go up further, 1 further, all the way up to G-gamma 13. Okay, so those are all the 12 different gamma genes which will code for a single subunit, and their subunits will be named after the same thing as the gene, basically. Okay, so that gives us an appreciation for the scope of different heterotrimeric G proteins which we have. Okay, we now know that we have 21 different alpha subunits, 6 different beta subunits, and 12 different gamma subunits. Okay, right. So what I now want to discuss is the G protein cycle, which is how heterotrimeric G proteins can be activated by G protein coupled receptors. And then what we'll do is we'll move on to specifically which heterotrimeric G proteins are going to be activated by these vasoactive intestinal peptide and pituitary adenylate cyclase activating protein receptors. Okay, so the G protein cycle then. So we need to start with our G-protein coupled receptor and our heterotrimeric G-protein. Okay, so here is our G-protein coupled receptor with its characteristic 7 membrane-spanning alpha helices. Here's the carboxylic acid terminus in the cytoplasm, and here's the amino terminus extracellularly. Okay, right, so this is our GPCR, and let's say the ligand is going to come in. Now we know that, in fact actually, oh well, I can't change that picture now, but I would have preferred, since we're talking about the VIP and PACAP receptors, to have drawn one that looks more like a secretin family with a larger amino terminal domain, but never mind. Okay, we'll just keep it general at the moment. So, the G-protein cycle then. Let's say our ligand is going to come in and activate the G-protein coupled receptor. Okay, now before the ligand has come in, we'll have our inactive heterotrimeric G-protein sitting here. So I'll just show this. So here is our alpha subunit with GTP, sorry, not GTP, that's very bad, GDP, guanosine diphosphate bound, so it's in the off state, and then it's got this beta-gamma complex attached to it over here. So here's the beta subunit, here's the gamma subunit, and remember the gamma subunit has that geranyl group holding it in the phospholipid bilayer, like so. Okay, right. So we'll colour everything in. So here's the gamma subunit in green, and here's the alpha subunit in red. Now, basically when the ligand comes in and activates the G-protein coupled receptor, what will happen is you'll get a conformational change in the G-protein coupled receptor that will be translated down to these free intracellular loops here. Okay, the free intracellular loops will change conformation and they'll make available a binding site for the alpha subunit of heterotrimeric G-proteins. Now, different G-protein coupled receptors will have different alpha subunits that they want to bind to. Okay, so for instance, this GPCR will have certain alpha subunits that it likes to bind to, and it will therefore only bind heterotrimeric G proteins if they have a certain alpha subunit. Now, this is the reason that when we name heterotrimeric G proteins, we usually label them according to which alpha subunit they have. Okay, so I might just have a little discussion of naming of heterotrimeric G proteins. So, ideally, in the ideal world, when we're dealing with a heterotrimeric G protein, it would be nice if someone labelled up exactly which alpha subunit, exactly which beta subunit, and exactly which gamma subunit. So let me give you an example. G alpha I1, beta 1, gamma 1 would tell us that we're dealing with an alpha subunit that's an alpha I1, a beta subunit that is beta 1, and a gamma subunit that is gamma 1. People rarely do this. The reason is that firstly they don't know, and secondly it doesn't really seem that important to know this sort of information. Okay, because as far as the signaling cascade that it induces is concerned, which beta and which gamma subunit you have doesn't seem to make a huge amount of difference, at least at our current level of knowledge. Now that is probably ridiculously naive to think that which beta and which gamma subunit you have doesn't really make a difference. Okay, but at the moment, that's our current position, that beta and gamma don't really make that much difference. Okay, so all we really care about is the alpha subunit. Now, firstly, people generally don't even tell you exactly which alpha subunit you're dealing with. They give you an idea, okay? And really that's because it doesn't make sense to say that you're specifically, well, rarely does it ever make sense to say that you're specifically activating a heterotrimeric G protein of the G with an alpha subunit that is G alpha I1, okay? Because generally G protein coupled receptors which activate heterotrimeric G proteins with a G alpha I1 subunit within them will also activate heterotrimeric G proteins which have other G alpha I subunits in, so G alpha I2, G alpha I3. So it doesn't really make that much sense to specify to this level of detail that this is the heterotrimeric G protein that is going to be activated. So instead, people will often just call, for instance, this a GI heterotrimeric G protein to tell you that the alpha subunit is one of the I's, okay? Alternatively, they might not even specify that much. They might say GI/0, and what they mean by that is it's one of the I, so it could be G-alpha I1, G-alpha I2, or G-alpha I3, or it could be G-alpha 0, because that does pretty much the same thing again, or potentially also G-alpha Z, which also does a very similar thing to those other four. Okay, so often you will find people labeling the heterotrimeric G protein according to which alpha subunit you have, and generally they'll only give you a sort of idea of what alpha subunit it is. So for instance, another example is people will often put GQ/11 to say that we're dealing with a heterotrimeric G protein where the alpha subunit is within that family of G-alpha Q/11 family alpha subunits. Okay, so it could be G-alpha Q, G-alpha 11, G-alpha 14, or G-alpha 15/16, because they all pretty much do the same thing. Okay, and often G-protein coupled receptors which activate one will activate the others as well. Okay, right. In addition, people often put G12/13 to say that the alpha subunit of this heterotrimeric G protein is either G-alpha 12 or G-alpha 13. Okay, generally when people put something like GS, you can assume that the alpha subunit of that heterotrimeric G protein is actually G-alpha S. If they mean G-alpha olf, they will put G-olf there. Okay, and that's very rare when they'll talk about G-olf. Unless you're looking at the olfactory transduction pathways, you won't really see G-olf that much. Okay, and of course G-alpha S has many splice variants, so they won't tell you specifically which splice variant. They'll mean one of the splice variants, or potentially it's activating all of them, because they are all very, very similar. Okay, so that's just a little bit on the naming of heterotrimeric G proteins. Okay, right. The G protein coupled receptor is going to become activated by the binding of the ligand. It's going to change the conformation of its free intracellular loops, and its free intracellular loops will change conformation to make available a binding site for the alpha subunit of the heterotrimeric G protein. Now, as discussed, this binding site will have affinity for certain alpha subunits, but not all of them. Okay, so it will have its favorites, and generally it will activate alpha subunits that are similar, and therefore they'll most likely have similar targets as well, because, you know, if there's similar structure enough to bind to the same G protein coupled receptor, then they're probably going to be similar enough to act on the same targets. Okay, right. So, let's draw the next stage then. So the alpha subunit is going to bind to our G protein coupled receptor intracellular loop region. Okay, so I'm just going to spin the G protein coupled receptor around 180 degrees, okay, simply because I don't want this carboxylic acid domain to be in the way of me showing the alpha subunit bound to the intracellular loops. Okay, so I've just spun my G protein coupled receptor around like that. That's just the basis of the drawing. Okay, right, and now in comes my alpha subunit here. Okay, still attached via this palmitoyl or myristoyl group to the inner leaflet of the phospholipid bilayer. And the alpha subunit will now have guanosine diphosphate bound to it. So here it is. Okay, right, so let's colour this in red again. Oops, smudging slightly, but never mind. Okay, now, the beta-gamma complex doesn't itself bind to the G protein coupled receptor. However, it is absolutely essential for the binding of the alpha subunit to the G protein coupled receptor intracellular loops that you have a beta-gamma complex bound to that alpha subunit. Okay, so to put it another way, if the alpha subunit did not have a beta-gamma complex bound to it, it would not be able to bind to the intracellular loops of the G protein coupled receptor. So for this interaction here to take place, it has to have this bit sticking off the alpha subunit. Okay, and of course, to have this bit sticking off the alpha subunit, you need the alpha subunit to be bound to GDP, to be in the off state. Okay, right. Now what's going to happen is once the alpha subunit is bound to the intracellular loops of the G protein coupled receptor, the alpha subunit will give up guanosine diphosphate, okay, and it will accept GTP from the cytoplasm. Okay, so binding to the G-protein coupled receptor catalyzes this conversion. This conversion happens ridiculously slowly if you are not bound to the G-protein coupled receptor. The reason is that when you remove the guanosine diphosphate temporarily, you have an intermediate where the alpha subunit has nothing bound to it. Now, that intermediate is incredibly unstable, basically. Okay, it's incredibly high energy. So when you wrench the GDP off, you have to then temporarily go to an intermediate that's got a high energy. Okay, so the activation energy for the removal of the guanosine diphosphate is very high when the alpha subunit is not bound to the G-protein coupled receptor. So to put this another way, if we draw an energy diagram, okay, this is the GDP bound form here. Okay, to go to the form where you have nothing, there's a very high energy barrier, and then you can come back down to the form where you have guanosine triphosphate bound. Okay, but you're very unlikely to get this, this activation energy here. So when the alpha subunit is just on its own, it's very unlikely to undergo this change. Whereas when the alpha subunit binds to the intracellular loops of the G-protein coupled receptor, effectively it brings the activation energy right down, so that this temporary alpha subunit, which has nothing bound here, is much lower energy and much more obtainable. Okay, and that's how you get the catalysis of this conversion of the alpha subunit from the off state into the on state. Okay, so the alpha subunit then has GTP bound to it, and when the alpha subunit gets GTP bound, what happens is it no longer wants to bind to the beta-gamma complex, so the beta-gamma complex cleaves off the alpha subunit, okay, and when the beta-gamma complex cleaves off the alpha subunit can no longer bind to the intracellular loops of the G-protein coupled receptor. Remember, I told you that for the alpha subunit to bind here, it was essential for it to have the beta-gamma complex bound as well. Now it's lost the beta-gamma complex because it's got GTP bound to it, so it's also going to cleave away from the intracellular loops of the G-protein coupled receptor. So let me show you what we've now got. So I don't need to draw the G-protein coupled receptor anymore because neither of these things is bound to the G-protein coupled receptor anymore. So here is our alpha subunit here, which is now in the on state with GTP bound and is floating around underneath the inner leaflet of the phospholipid bilayer. And also we have the beta-gamma complex, which is also now on its own, detached from the alpha subunit. Okay, so here's the beta subunit and here's the gamma subunit, and again they have gone off on their own. So I'll colour in gamma in green here, and we'll have beta in blue. Right, and then we'll colour alpha in red as always. So now what's going to happen is each of these is going to go off and interact with targets, okay, and the targets will differ depending on which type of alpha subunit you have here, okay. As far as we know at the moment, it doesn't really matter what beta and gamma which beta and which gamma subunit you have here as far as the signaling of the beta-gamma complex is concerned. But of course that's ridiculously naive, and that will of course change, most likely anyway. So we have a target here for the beta-gamma complex, so let's call this target 2, and then principally the thing that everyone's generally interested in are the targets for the alpha subunit. Okay, so we'll call this target 1. So the alpha subunit will go off and bind to its target and will affect this target's function in some way, and the beta-gamma complex will bind to targets as well and affect the function of this target. And as I say, we most definitely know that the type of alpha subunit you have here affects which targets it interacts with and how it affects them. Okay, but with regards to how the different beta-gamma complexes interact with different targets, that's still very little is known about that. Okay, right, so the alpha subunit will bind to its target, cause some change, okay? But at the same time, when the alpha subunit binds to its target, not only does the alpha subunit affect the target, but also the target is going to affect the alpha subunit, okay? Specifically, it's going to activate the GTPase activity of the alpha subunit. Okay, so the alpha subunit is actually an enzyme. It's an enzyme known as a GTPase, which means that it catalyzes the hydrolysis of guanosine triphosphate into guanosine diphosphate, GDP, and inorganic phosphate. Okay, now it's not usually particularly active, but as soon as it binds to its target, and I would like to colour its target in here, so let's have the target in turquoise here. As soon as it binds to its target, the target will activate the GTPase activity of the alpha subunit, and it will start to hydrolyze the GTP into GDP and inorganic phosphate. Now, the inorganic phosphate will be released into the cytoplasm, whilst the GDP will remain bound to the alpha subunit. So effectively, target 1 causes the alpha subunit to turn itself off. It causes it to go from being in the on state to being in the off state. Okay, now once the alpha subunit has turned itself off and has guanosine diphosphate linked to it, it will go off. It will detach from target 1 because it can only bind to target 1 when the alpha subunit is in the on state. So as soon as it's turned itself off, it detaches from its target, and then it runs off crying to the beta-gamma complex, basically, snatches the beta-gamma complex off its target, and then they reform the inactive heterotrimeric Gprotein, and that's why it's called a cycle, basically, because as you can see, we've gone round here. So this is the Gprotein cycle, the cycle of activation and then self-inactivation, okay, of the heterotrimeric G proteins. Right, so what we now want to turn our attention to is which specific alpha subunits are going to be activated by the vasoactive intestinal peptide and pituitary adenylate cyclase-activating protein receptors, and what are the downstream targets of those heterotrimeric G proteins. Okay, but we'll do that in the next video.